This setup is a little bit strange, a little bit different from normal, so forgive me if I have to like adjust in the middle of the review. There should be a bunch of sign-in sheets going around. I'm going to hand out a few more. You only have to sign one of them once, so make sure you keep passing it around. I'm not allowed to see the file, but yeah, I'm taking this file. Let's see. Where did I put it? So before we go on, I'm sure some of you got an email from the TA, right? She's talked about some exam review that she's going to host on Saturday. So what she's going to do Saturday from 8 to 9 is allow you to review exam 3. So you can go in there and see what you did on exam 3, see what you got right, see what you got wrong. Note that you're not allowed to take notes during that session. However, since the final, 70% of the final is going to be on exam 3, plus the translation chapter, I highly suggest you go to that review session. Another thing is that Hasey's has told your TA to do her own final exam review as well. That will also take place on Saturday from 9 to 11 a.m. She hasn't sent like an updated email or anything, right?
No.
Yeah, so unless something changes tonight, it will be on Saturday from 9 to 11, right after the exam 3 review. During that review, she's going to go over questions and concepts that are going to be on the final, so I highly suggest you go through that as well. Okay. This exam review, we're mostly just going to focus on exam 3 material, chapter 30, and a few things from the first 2 exams that he said were going to be important. We don't have enough time to cover everything from exam 1 and exam 2, but we'll see how far we can get today. So, first, we should start with chapter 30. So we're going to do the usual format. I'm going to go through the slides for the only unit that we haven't done this for yet, and I'm going to say what's important, what's not important. Then we're going to go over more specific stuff. Okay? So, let's see. So, referring to the codon, you need to know the features of the triplet code. You need to know why we need a triplet code and not a doublet code or quartet code. Does anyone remember why we need a triplet code? What is that feature? There's 20 amino acids, right? How many bases do we have? 4. Right? We use 4 nitrogenous bases. So every possible combination we can make with 2 bases only adds up to 16 total combinations. There are 20 amino acids, so we must be able to generate 20 or more combinations. That's why we use a triplet code, because you'll see in the practice exam that he has a question like that where you have to calculate how many combinations you get with a triplet code or a quartet code. A triplet code, you get 64 possible combinations. That's plenty. That's over 20. That is why we use a triplet code. Let's see. You don't need to know history stuff, so whenever they talk about history or how they discovered something, don't worry about it. Let's see. You do need to know the different areas of the tRNA, so you need to know what the acceptor stem is, right here. What is this area up here where it says CCA? What is that important about that area? That's where your amino acid is loaded. Very good. So that's one area that's important. The place where what's the part of the tRNA that binds to the mRNA? The anticodon region, right? This 3-base sequence is going to look for a complementary codon on the mRNA. Okay, so you need to know the anticodon region, the acceptor stem, and there's a few other ones we'll get to when we talk about recognition of sequences.
What is the anticodon that's already
more history stuff you don't need to know. And here's the calculation, by the way. So you do need to know what they mean when they say is the code overlapping or punctuated. So they give a diagram right here where they talk where if our code was overlapping, that means the first 3 bases generate 1 amino acid. But then they reuse base number 2 and 3 and just add on base number 4 to generate the second amino acid. We know that is not how translation works, so you need to be able to identify that translation is non-overlapping. Okay, that's a characteristic you need. In terms of whether the code is punctuated or not, they also diagram that here. They say if the code was punctuated, the first 3 bases generate 1 amino acid, then there is a base that we don't use at all, and then they use the next 3. We also know that is not how translation works. So you need to know that the triplet code is non-overlapping, non-punctuated. What do they mean when they say the code is universal? Every organism uses the same code. AUG codes for methionine in bacteria and eukaryotes in all sorts of organisms. So you need to know those key vocabulary terms. What do they mean when they say the code is degenerate? In other words, there's only 20 amino acids, but there's 64 combinations of codons. That means multiple codons can code for a single amino acid. That's what the term degenerate means, and you need to know that. Let's see. You don't need to know how they figured out that, so skip that slide where it says poly A, all that stuff, you don't need it. He says skip that. So, if you are given this table, it will be for a question where he gives you an mRNA sequence and you need to give him the corresponding amino acid sequence. So you need to be able to learn how to use this table. This table is organized by codons and their corresponding amino acids, not anticodons. So you read your mRNA sequence, you look for the codon on this table. What must be the first codon when you do translation?
AUG.
AUG. So make sure if he's that tricky that he gives you a bunch of other amino acids, before, a bunch of other codons before AUG, you do not use those. Okay. You also need to be able to identify the stop codons as also indicated on this table. How many stop codons are there?
3.
3. Very good. Let's see. And once again, you don't need to know the table. He will give it to you. Okay, talking about the next step is, we know that tRNA is the molecule that brings the amino acid to the ribosome. But the tRNA doesn't come with the amino acid right away. Something attaches the amino acid to the tRNA. So you need to know what enzyme does that, and that's this thing here, amino acid tRNA synthetase. Now, how does amino acid tRNA synthetase recognize the correct tRNA for the correct amino acid? There are certain recognition sites, and let's talk about that here. I'll go back to the top in a second. You need to know the recognition sites for amino acid tRNA synthetase. You need to know primarily they're looking at the anticodon region of the tRNA. We remember that the anticodon is the one that makes it specific for an amino acid. Because of degeneracy, you can have multiple anticodons coding for the same amino acid. Okay. Now before we go on, we'll go back some more. You need to know other important activities of the amino acid tRNA synthetase. Not only is it the one that connects the amino acid to the tRNA, it is also the one that activates the tRNA for translation. Activates the tRNA for translation. Yeah. Let's see.
The Anti-coded universal. It's right there.
This one?
Well, the code is universal.
Yeah.
In the PowerPoint on the slide that says recognition of tRNAs, it says something about not universal. Can you clarify the difference?
Yeah.
That was there.
This
on
one?
the
Oh. Oh,
Anti-codon.
is this referring to the synthetase specifically? In other words, a synthetase in like a certain organism may not have the same recognition mechanism as a synthetase in another one. But yeah, the code is universal. Don't worry about this last line. This is too much detail. You don't need that. Let's see. Other important elements in recognition of the synthetase, you need to know these as well. So the synthetase not only is it looking for the anticodon region, it's also looking for some bases in the acceptor stem. So this is a good multiple choice question, like which of these is not used to recognize the correct tRNA. One base in the acceptor stem, a special base at position 73 called the discriminatory base, and we already mentioned the anticodon. Something special about the anticodon. Let's see if I can find it. Somewhere in your notes, they do mention that if the second base is a pyrimidine, it bonds to which type of amino acid, polar or non-polar? Polar, right? And if it's a purine, it bonds to non-polar, so you need to be able to identify that. Okay, so when you're looking through these, make sure you read what they do. Because remember, there was a practice question on that, so you need to be able to identify that. Let's see what else. This diagram is a nice summary of the different recognition sites. So you will not be asked to label a diagram for this, but this is a good organization. Acceptor stem, position 73, and the anticodon. Moving on. The wobble hypothesis, you in general need to know the concepts of wobble hypothesis. There was going to be a question on it, but remember that question in the practice exam that he talked about, question 16, that everyone didn't really like too much and he threw that out? That would have been the question he would have asked concerning the wobble hypothesis. That would have been his critical thinking question. So, if he still gives a question similar, it would be a little bit more simplified. He may give you this table here, and you would need to be able to use this table to base pair the wobble position. Which position is the wobble position on the codon? The first or the last one? The last one on the codon is the wobble position. The first one on the anticodon is the wobble position. Okay. So you may need to use this table to solve a question like that. Going back to the wobble hypothesis. Since there are only 20 amino acids, but 64 possible combinations, this kind of implies that not every base is important in identifying the correct amino acid. And we know through experiments that the first two bases are very specific for an amino acid, but the third one doesn't need to bind as specifically, right? And they use that term, they use the term non-canonical base pairs, so that's an important term you need to know. How many non-canonical base pairs are there per codon and anticodon interaction? Only one non-canonical base pair, because there's only one wobble position.
So are there two canonical base pairs?
Mm-hmm. And therefore, by inference, you need to know that there are two canonical base pairs per codon-anticodon interaction. Yes?
What's the significance of position 73?
So that's just a term for it. I don't know the exact theory behind it, but position 73, and what they mean by that is on this tRNA sequence, at position 73, it's called the discriminatory base. Depending on what that base is, helps determine what amino acid is going to be bound to the tRNA. You don't need to know specifically whether A means something, G means something, just a vocabulary term.
You said there were
two canonical base pairs, right, let me clarify. Two canonical base pairs per codon-anticodon interaction and one non-canonical base pair. The term non-canonical means it does not bind. It does not bind specifically. So we know normally A binds to T, G binds to C. The wobble position says, well, according to the table, that U can bind to A, G, or I, G can bind to C or U, etc., etc. So if he gives a problem like that, he would need to give you the table. It's more likely to ask conceptual questions about the wobble hypothesis rather than one where you have to sequence it. Let's see. Talk about this. So we know ribosomes are made of two subunits, and what two components together? So ignoring the subunits, what molecules are involved in the ribosome? Protein and RNA. Protein and rRNA, correct. So you need to know it's mostly which one? Mostly protein or mostly rRNA? Mostly rRNA, 2/3 of it. You don't have to remember whether 23S means something or 5. You don't have to remember the numbers. If you see a larger number, like 80, that means your large subunit. If you see a smaller number like 20, that's your small subunit. That's the furthest extent he's going to require you to know which is your large and which one's your small subunit. You need to know where translation occurs or where you can find ribosomes. And in prokaryotes, where's the only place you can find ribosomes? In the cytosol, right? In eukaryotes, you can find them in the cytosol, you can find them in mitochondria and chloroplast. Why do you think mitochondria and chloroplast have their own ribosomes?
Because they're prokaryotes initially.
Because due to evolutionary theory, we have figured out that mitochondria and chloroplasts likely originate from prokaryotic organisms, so they have their own unique ribosomes, so they do their own translation. More diagram stuff you don't have to worry about. And more stuff about the ribosomal numbers, like L20 and stuff, you don't have to worry about. I wish I got a table or something like before.
You can grab that podium. Podium. We can help you drag it over here.
He said we didn't have to do this.
Alright, so we'll go on to the next one.
I'll help you drag it. Okay.
He said we didn't have to.
So this one I'm going to simplify into, you know that he gave you this video, right? Definitely study this video. It is most of this PowerPoint. There's a few details that are not covered in this animation. For example, the difference between prokaryotic and eukaryotic translation. There is a slide that details 6 differences between prokaryotic and eukaryotic translation, and you need to know all those differences. We'll get to that in a second. Okay. So actually, one of the differences has to do with initiation of translation. In prokaryotes, we require a special type of methionine called formal methionine, right? There's a few questions you can ask on that. One is, how many tRNAs are needed for the initiation of methionine in prokaryotes? Only one tRNA in prokaryotes. So this is a little bit tricky. We'll talk about it even more. The formal methionine in prokaryotes, where does the formal come from? There's an enzyme that specifically edits the methionine to make it formal methionine. So it's the same tRNA, but another enzyme comes in to edit the methionine to become formal methionine. This is talking about prokaryotes. Is there formal methionine in eukaryotes?
No.
There's no formal methionine in eukaryotes involved in initiation. So you need to know that formal transferase is the enzyme that adds the formal component to the methionine. You need to know that the formal part of the methionine doesn't necessarily stay on the methionine when you finish your peptide. Sometimes the formal is removed with the enzyme called deformylase. More pictures. There is a question, actually it's on the practice questions that I have, so we'll go over that later. Prokaryotes and eukaryotes, which one has Shine-Dalgarno sequences?
Prokaryotes.
Only prokaryotes. And what is the Shine-Delgado sequence used for? That's where your ribosomal subunit is going to bind to. So in prokaryotes, before you can begin translation during the initiation phase, the small ribosomal subunit is looking for the Shine-Delgado sequence to bind to. Okay. In eukaryotes, there is no Shine-Delgado sequence to detect it. In general, in eukaryotes, the small subunit is just going to bind to the mRNA near the G' cap. We remember that in eukaryotes, there's some editing to the mRNA, yes?
Yeah, what's the Shine-Delgado sequence? What is it attached to again?
So the Shine-Delgado sequence is on the mRNA, and that's the place that your ribosome is going to attach to in prokaryotes. Because in translation, we need the ribosome to bind somewhere, and it prefers to bind to the Shine-Delgado sequence. In eukaryotes, there is no Shine-Delgado sequence, so how does it know where to start translation? Well, in eukaryotes, ribosomes prefer to bind near the G' cap of the mRNA. If you don't remember what the G' cap is, or the poly A tail, one of these PowerPoints, I think, in the transcription section at the very end of it, they talk about splicing of your mRNA. He says you don't need to know a majority of the splicing stuff, but you need to know the basics. So stepping back a little bit, in eukaryotic mRNA, the types of editing you need to do is you need to remove what in your mRNA?
Introns.
Introns, that's one type of splicing. Another one is you need to add a poly A tail and a G' cap to your mRNA. All of those features are only in eukaryotes because you are editing mRNA in eukaryotes. Going back to translation, in eukaryotes, you're looking for the G' cap, you attach the mRNA there, and you move along the mRNA until you read the first start codon, which is A to G, right?
A picture.
You don't need to know what a Shine-Delgado sequence looks like, whether it has like certain numbers of G's or C's or T's. Okay. Talking about initiation, you need to know all the materials required for initiation and translation. We recognize we need the mRNA. We recognize, actually, which subunit does the mRNA initially bind to?
The small.
The small one, that's very important. It initially binds to the small subunit. You also need initiation factors. You don't need to know whether 1 does what, 2 does what, or 3 does what. You just need to know that there are 3 initiation factors. Okay, so you need the mRNA, you need the small subunit. You will eventually need the large subunit. So if we're making the list, we can put that on the list. You need the initiation factors. You need energy. What provides the energy for translation? GTP. Very good. And for prokaryotes, you need a special type of tRNA amino acid bond, and that is the formal methionine bonded to the tRNA. Remember that it's technically the same tRNA as regular methionine, but this methionine happened to be edited to include a formalin. Okay. Let's see. So before the large subunit can come in, you must remember that the initiation factors must be removed. Okay. So he may be specific and he asks you, when can the large subunit come in, so make sure you know that. In terms of the experiment or anything, you don't need to know how they found this out, but you need to know what is italicized here. That if you have in your sequence the same amino acid being repeated over and over again, usually it's the same codon that's saying that, that's repeating that amino acid, and it's usually the same tRNA that's bringing that amino acid. Okay. Everything else, like how they found that out, don't worry about it. Okay. Now talking about elongation, we recognize that there are 3 elongation factors, right? You need to know what each one does. There's F2, FTS, and what's the last one? So you need to know that F2 is the one that does what? F2 is the one that brings the tRNA and the amino acid and activates it for activates the peptidotransferase activity of which subunit? Which subunit has the peptidotransferase activity, the small or the large one? The large one. It's another thing you need to know. So if F2 is the one that's using GTP, what factor is the one that generates that GTP? FTS. So this is a repeating procedure. Remember that elongation must be repeated several times for each peptide, for each addition of a peptide in translation. So you need to know each step. I won't go over any more of that. It is in the animation for you. Termination is actually really simple. You need to know that you read a stop codon, but what binds to the stop codon?
The anticodon.
So that's actually a trick question. There is no tRNA or anticodon for the stop codon. Release factors actually bind to the stop codon, and you need to know that. So there was a true-false question either on the practice exam or something where he says there are tRNAs that bind to the stop codon. You know that's false. Let's see.
You never get true or false questions on this exam.
When the release factors are there, they cause the peptidyltransferase to instead have hydrolase activity. So it causes everything to dissociate from each other. So on. Of this antibiotic selenium slide, you need to know how in prokaryotes, how there's a special tRNA for selenocysteine and how that is formed. It is detailed in this slide, so I'll leave that to you. Okay, this slide and this slide is where they compare the prokaryotic and eukaryotic translation. You need to know everything on this one, and according to him, you can skip number 4 here. Okay. Everything else is fair game. So we already talked about that in prokaryotes, you need to have formal methionine to initiate translation. How do eukaryotes tell the difference between initiation methionine and internal methionine? There's a special tRNA used for the initiation methionine and a different tRNA used for the internal methionine. So that could be a question you ask. In general, we know that eukaryotes have larger ribosomes, correct? And we also know that eukaryotes do not have Scheindel-Garno sequences, and so on and so on. I'll let you guys look at the rest. So he said you can skip everything else in eukaryotic translation. Okay. All right, so that was the usual stuff. Now we're going to talk about some practice.
People wouldn't know.
And for exam 1, so remember that exam 3 is 70%. Exam 3 plus chapter 30 is 70% of your final. And there are still going to be some stuff from exam 1 and 2. The practice final, yeah, I'm going to send this to everyone after this. I just made this like today, and it's very, very up to date.
Did you save it already to Blackboard?
No, I have to, this is like a rough draft. I have to edit it and send it to everybody. It's nothing, okay, so if you're wondering if this is like really, really like crucial stuff, it's just a few true-false questions and fill in the blanks. It's nothing like multiple choice or anything. There are a few multiple choice on here. But before I forget, remember the TA is having her own review on Saturday. She will be providing questions and concepts that are going to look similar to the final. But when we go over this, I'm going to skip down to the translation stuff and we'll go back to the other things. All right, so we'll talk about the translation stuff, then we'll go back and talk about replication and transcription. So, when we're talking about characteristics of the genetic code, this is sort of like pick which is correct, and it's a good one to recognize which is true and which one's false. How many codons signal the start of translation? Only one start codon, so A is incorrect. B, there are 3 codons that do not code for any amino acid. That is true. That's what your stop codons are. They do not code for amino acids, so that one's your true answer. Most amino acids have only one codon we know is false due to what vocabulary word they use to describe the triplet code? Degeneracy. Very good. codes for amino acids differ in prokaryotes and eukaryotes.
False.
False because of another vocabulary word. The triple code is universal. Okay? Next one, a tRNA molecule recognizes a codon through what? So this one is a little bit tricky. We know primarily it recognizes it through the anticodon and codon interaction. However, if you look at this choice, notice that I say the anticodon sequence is from 5' to 3', while the codon is from 3' to 5'. Is that correct?
Nope.
It's the other way around. Remember that mRNA, when it's going through translation, is being read from the 5' to 3' direction. So if B was flipped, if they flipped those numbers, that would be correct. In translation, are covalent interactions involved? No, non-covalent interactions, more specifically hydrogen bonds, right? So we know C is incorrect. The extra arm variable length, that is one of the recognition sites, not for tRNA, but for the synthetase, right? That was on that list of recognition sites for the synthetase, not for when the tRNA is recognizing a codon. So, our only one here is either D or E. We know D, where they're talking about the anticodon having 3 bases complementary to a codon. That is correct. So the answer there is D. Okay. Which of the following statements are true? A group of 3 amino acids code for proteins.
False.
That's false. The amino acid found at the starting point of translation is tryptophan. We know that's false. It's supposed to be methionine, right? Usually several codons can designate one amino acid. That's true, that's degeneracy, so we know that is true. And the specific binding of amino acid tRNA is not directed by trinucleotides bound to ribosomes. That's a complicated way of saying how does the amino acid tRNA know when to bring what is it specific for? It's specific for the codon. Not really specific for the trinucleotides bound to the ribosome, but specific for the bases on the codon. Okay, so we know D is incorrect because it's not phrased correctly. Which is not true regarding the activation of amino acids and amino acid tRNA synthetases. Okay, remember that the synthetases recognize certain things. It recognizes the anticodon. It activates the amino acid for transfer. On your slides, they mentioned a few other activities. They mentioned deacylase activity. So if you go in there, you'll see it details that deacylase corrects the synthetase. In case the synthetase put the wrong amino acid on the tRNA, that is used to correct that. Once formed tRNA can have more recognition sites besides the anticodon region. That's not true. So D should be the answer. Once formed. So before we know that there's many recognition sites, the discriminatory base, the anticodon region, and the acceptor step.
Are you putting the right answers to this?
Yeah, this is one thing. So